Thank you very much. Yes, first of all, the title here, using epidemiology data towards causal inference, is rather provocative. In toxicology, it's very much an opinion, a broad opinion, that epidemiology can be used for causal inference because you are measuring on real life, so to say, and there is a lot of background factors that can introduce bias into what you are looking upon. And the only thing that we can trust on is experimental toxicology when you are working in cells and animal models, for example. What I'm going to talk about today is what we are doing in environmental epidemiology, but where we also have in recent year, I would say, start to come a little bit more towards this causal inference. And my ambition with this is that epidemiology should be used much more in risk assessment. As it looks today, it is almost not used at all, which is both very strange, but also very sad, I think. So as you can see from this picture, we are working with humans, and we are very interested in the early life during pregnancy, and what happens with children in a very broad context. So what I'm going to talk about today is I will introduce you to this SELMA study, which is a pregnancy cohort, meaning that we recruited pregnant women and are following these families, and the children to these pregnant women are today around 12 to 13 years old. I'm going to come into a very complicated area, namely the difference between single compounds and compounds in complicated mixtures. This is a big question today, how all risk assessment today is about single compounds. But everyone knows that we are never, ever exposed to single compounds. We are always exposed to mixtures of compounds. And this is a challenge. And we have been working quite a lot with this. And my third theme here is to show you some ideas we have with SELMA as a basis for development of environment epidemiology towards more causal inference. And I will come back to that more. So first of all, you can raise your hand if you or write in the chat, but I can also, I'm comfortable if you ask directly, but we can have the discussion maybe after. But if there are things that you don't understand, please remind me so I can explain better. So SELMA is a matter of it's a pregnancy cohort, as I said, and it was started in the year of 2007. And
Gustav, there is actually a question already now. So from
Yeah, okay. So actually just because of curiosity, but you did this in Värmland. You follow people in Värmland.
Yes.
Why is that, if I can ask?
Well, I live in Värmland.
Okay.
No, but yes, I mean, it's partly true. But there are also other opportunities for us to do it in Värmland, because we have had a very close, Karlstad University have been working very close together with the former county council and what today is the region of Värmland, the healthcare system. And very much of the data collection in a study like SELMA is much more effective if you can integrate that in childhood health care or mother health care, and that is the infrastructure for collecting data is there. And in Värmland we have had a very, very good collaboration with the county council. So that's one of the reasons. Okay, so the overall aim here is with the SELMA is to look upon early life, and with early life, I mean beginning of pregnancy. We are, I mean, the most important thing we are looking upon is chemicals with proven or suspected endocrine properties. That is chemicals that reminds of natural hormones, and they may disturb very important developmental phases during fetal development. And the point with this, and which make it very complicated, is that even if you have different kind of programming effects during the fetal development, maybe the outcome of this or the effect of this is not seen, will be obvious much later on. It is also a possibility that some of these effects are seen generations downstream. So this is a very complicated story about this kind of focus. Very much of this environmental epidemiology is coming from occupational settings where you have a high exposure and you have the effect much more related to the exposure. Meanwhile, this is not about toxicity. This is much more about programming effects. And our interest is, we have measured a lot in the blood and the urine of the pregnant mothers. And then we are following the children, and we are looking in 4 domains, so to say, today. One is growth and development, birth weight and growth during childhood. Another one is sexual development. It's about brain development or neurodevelopment, and it's about asthma and allergy. And I will show you some of these results, but this is the overall design of what we are doing. So one of the first scientific focus in this area of endocrine-disrupting chemicals was some 25 years ago, about sexual development. It was where it all started. And the reason for that is that several of these chemicals we are very interested in, they do have properties of androgen and anti-androgen effects, meaning that they can regulate the human balance of hormones in a way. And this is very much a matter of estrogen and testosterone. And so this was where I would say, started some years ago when we start to get the results. So we have measured these anti-androgens in the mother during week 10 of pregnancy. And here is a big focus have been on phthalates. Phthalates are found in plastics and especially in soft plastics, the rubber duck, but a huge number of products are including phthalates. And it has been shown earlier that these kind of chemicals do have properties that can downregulate testosterone, for example, and upregulate estrogen, and that have impact on sexual development. So we are looking in this area in two domains. First of all, we have measured what is called anogenital distance. That it's a measure in baby boys when they were around 21, 22 months old. And this is known since 15 years. You see a picture of Shanna Swan here from Icahn School of Medicine at Mount Sinai. I've been working with her for quite a long time, and she is a pioneer in this area. And this measure is telling it's a marker of the androgen status during early pregnancy. And this is also, if this is changed of some reason by the environment, it can be interpreted in terms of feminization, meaning that you are moving on a scale from male to female status. We are also looking here, and this has been starting right now with a PhD student connected to Uppsala University, on play behavior, this PS AI, which is a scale for typical female and male play behavior. And we are very interested in to see if we, by environmental exposures, can change this behavior. And we have findings pointing in that direction. So to make a very long journey short, this is 5 different phthalates in mother's urine in week 10 of pregnancy. And what you can see here in odds ratio, it's the risk of having a shorter anogenital distance. It's the distance from the anus to the scrotum in baby boys. And what this picture shows that for one of these phthalates, or maybe 2, the DHP and the DINP, we can see an association telling that the higher exposure, the higher levels of these 2 compounds in the mother's urine in week 10 of pregnancy, the shorter is the distance from the anus to the scrotum in the baby boys. So, okay, this is what we see. But then the next question is, of course, how to interpret this. And this is rather difficult, but the anogenital distance is a matter of sexual development. And there is quite a number of papers that are interpreting this as kind of a feminization. This is published and you can read about it. And we are right now in the area of this play behavior. And here, of course, we are very interested to see if these kind of environmental exposures can have impact on especially the male play behavior. So if they, in terms of their genitals, are moving more to a female side, is it also possible that they change play behavior? And we have some promising findings here, but this is just under the way. So another part here is also neurodevelopment. It's the same thing. We are interested in several of these compounds that we have measured in early pregnancy and what they do in terms of neurodevelopment. And here it's a very important thing to remember that if you have chemicals that have these anti-androgen properties, the first you think about is, of course, sexual development. But we should also know for sure that, that especially testosterone is a very important hormone for brain development. So there is a lot of reasons to believe that if you see an effect from several of these compounds on sexual development, you can see it also on neurodevelopment. And here we have measured quite a lot. We looked for language delay in the children when they were about 2.5 years old. And at 7 years, we made psychological tests. And here we have used the WISC scale, which is about cognition, the IQ of the kid. And right now we are analyzing data in this STQ scale, which is more about behavior and also related to ADHD and these psychiatric disorders that you know about. So I just give you one slide here. Bisphenols, they are another group than the phthalates. The BPA, I think you all have heard about BPA, bisphenol A. It has been discussed for decades. This is not from soft plastics, it's from hard plastics. And this have been discussed very much. And a lot of opinions have told that we should try to get rid of this. Right now, the limit values of the intake of bisphenol A is dramatically decreased. So I think the authorities around the world have been starting to take this seriously. The problem is then that the industry is replacing BPA with other compounds. So here you can see a slide of BPS or BPF or 45, 50 others. So this is really what's going on out there. When a chemical is too hard to produce, of these kind of opinions, then you can replace it with another one that have the same properties. And this is really frustrating. We have looked on this bisphenol A, and here you can see a slide in a quite recent publication where we could see a continuous association that the higher concentration of bisphenol A in mother's urine in week 10 of pregnancy, the lower cognitive function, the lower IQ in the children. And the association were stronger for boys than for girls. This is, I will come back to this a little bit later, but this is, you will see here in many of my result presentations that we most often see different associations for boys and girls. And this is basically explained or hypothesized that if this is a matter of disturbance of the hormonal balance, then this is quite reasonable that you have different effects for boys and girls. And you should, for example, testosterone during early fetal sexual development. Testosterone is essential, very important for a proper sexual and genital development for the boys, but it doesn't matter for the girls. We are all girls from the very beginning, and the boys need the testosterone during that phase, that window of time, but not the girls. For brain development, there is no difference. So then I come into the second part of this, which is a more, I would say, a more important part of it. Just to guide you and to give you an introduction here, how do we do risk assessment? This is the way we have done this for 100 years. We know something about the human exposure. We are using what we call biomonitoring data. It could be blood, urine, or different kind of samples, hair or saliva or whatever it is. We know something about the human exposure. And of some reason, we have suspected that this might be a risk. So then we go into experimentals. We use animals. Nowadays, we are trying to move from animals over to cell system. But the point is that we are exposing we are dosing and we are looking for effects. And this is then resulting in what we often call reference doses. That is a dose for which below that we don't see any effect, but above that you can see an effect. And then we go back to the human situation and put up what we call the regulatory ratio. And that is the human exposure in relation to the reference dose. And you can imagine here that if the human exposure divided by the reference dose is higher than 1, then we are in problem, so to say. But in the opposite, if it's lower than 1, then we are on the safe ground. And then we put on some safety markers here. This is something that have been going on and have been very, very successful for 100 years. But the problem is that this approach, so to say, is based on that we take one chemical at a time. And that worked in history because this is coming from occupational settings. You are working in a dirty industry or in farming. You are handling dangerous and toxic chemicals. But the exposure is very, very high, and you can establish this approach and it works. The problem is this. This is from the SELMA study. It's measurements from 2,300 pregnant women in SELMA. We have looked in the urine and the serum from week 10 of pregnancy. And this is what we currently have. And in a few months from now, we'll double this list. But these are urinary-based compounds where we usually have metabolized compounds. It means that they are broken down and they leave us through the urine mainly. And in the bottom, you can see the serum compounds which are much more stable, and we call them, many of these are really problematic. You can think about the PCBs in the bottom. We did ban them for almost 50 years ago, and newborn kids today still have them have these compounds in their bodies. So these kind of chemicals are very, very different. But the point with this picture is the red column you see to the right. That is how many in percent of these 2,300 women could we identify each of the compounds we have here. And the point with this is that you can see here that almost all women are exposed to almost all these chemicals. So talking about single compounds in a risk assessment perspective doesn't work. We have to realize that we are exposed to very many at the same time. The problem here is that how to do that. And we have been working with this for 5, 6, 7 years now, that to come up with methods to look in epidemiology firstly. How can we estimate risks from mixtures of compounds instead of single compounds? And this is the same setup as I showed you before. We know the exposure in early pregnancy. We can look upon this as a number of chemicals, and if we can put a mixture approach here, that would be of importance for understanding the real exposure. And in this example, we are working with cognitive function, which is IQ of the children when they are 7 years of age. And Eva Tanner, you can see here to the left, she was at that time at Mount Sinai in New York City, and she has been working quite a lot of this and have a number of publications from the SELMA study. This is, I will make this quite short and talking about the results, but this WQS regression, weighted quantile sum, is a way to look upon the data where you look for association in a regression model as you do with single compounds, but you work with an WQS index, meaning that you have an index representing all the chemicals you have in the analyze. And what we could see here in 718 children, half girls, half boys, was that we could find an association, a significant association showing that there was a mixture of chemicals that was related to a lower IQ at age 7. And as I told you before, the association was stronger and more significant for boys compared with the girls. And you can see here, the scale here, difference in IQ score, is talking about that 1 unit in the exposure scale was related to an IQ loss of around 2 units on the scale. Which is quite big on a population level. And then the real question with this is, okay, but if you have a mixture that was of importance, which were the bad actors? Which were the chemicals that was driving this association? And here you have these 26 chemicals on the x-axis here. And you can see from the left, we have the urinary compounds, the phenols, the plasticizers, which is very much the phthalates, and other short-lived compounds. And to the right, then you have more these persistent compounds, the PFAS, called forever chemicals. They maybe never, ever will leave this planet because they are so extremely persistent. And the height here of the bars is the weight. And the interpretation of this picture, there was a mixture that was related to a lower IQ in the children. And that mixture of importance, the actors of importance, was looked upon to be BPF. You remember that from the former slide on, this is a bisphenol that have replaced BPA, and that was the strongest signal. But we have also signals from the phthalates. We have signals from other short-lived compounds, and we have signals from the persistent ones. And this is then a way to look upon a real life exposure. And the point is that here we can see associations that we, in many cases, can't identify when we looked upon them one by one. So another story that is of very, very great interest is to look on metabolism and growth. This has been, for me, a very an area of science that I've been extremely interested in, because one of the reasons is that we know for sure that how children grow and the birth weight, for example, is a predictor for the entire life in very many health aspects. It's not only about birth weight, it's also about the curve, the growth trajectory curve. And for those of you that have children, know that we put a lot of attention to the children's growth during early life. And there are reasons for that. And of course, growth is also a matter of the public health problem we have with higher body weights and BMI and these things. So the concept here is about the same. And the easy part of this is a result like this. This is Sverre Wikström, one of our MDs, and he looked on birth weight for the children, and he looked how much of these perfluorinated compounds, these forever chemicals, did the mother have in her serum during early pregnancy. And he could see a strong association, and you can see in this case, the strongest association was for the girls, but he could, for both sexes, he could see associations. So the higher levels in the mother, the lower birth weight. And this is, this is not, and you can see that if you look at the girls from low exposure levels up to the higher levels, it's around 100, 200 gram, which is quite a lot for a newborn baby. And here we are touching then another very amazing story. And I don't know much you know about David Barker, but if you have on Friday night after you have had a good dinner, try Google down some articles from David Barker, and I'll promise you will be amazed about this man and his science. He is dead. He died a few years ago, but he is the father of the so-called Barker hypothesis, because he realized in very, very old materials from the 18th century, from the 19th century, that a low birth weight, and the reason why he had such data was that he could go into church registers, because we have for very long time noted the birth weight of the children. And he could see that a low birth weight, when you control for as much as you can, is a risk factor for a lot of diseases 70 years later in life, like cardiovascular diseases, diabetes, and a lot of things that really kills us. So then what I showed you before here, the result, and no, I should say first, how did he explain this? Well, after a while, he realized that what this was about was starvation. So if the mother was under starvation, the kid, the baby, was born with a lower birth weight, and that was a risk for later on diseases. And he never thought about other environmental factors. But as you can see here, now we have shown that we can have environmental factors, namely these perfluorinated compounds, that gives exactly the same effect. And this is very much explaining in a way how these endocrine disruptors works. It's about programming during this early phase of life. Because what he realized later on was that if you were born with a low weight, then if you then increased in weight during early childhood, that was an even stronger risk factor for diseases later on in life. And this was his explanation of this later on, because he found materials, was that we are during fetal development. The old way of looking on fetal development is that the mother's only role is to protect the fetus. But he realized, and others have realized, that the mother have an even more important task, namely to program the fetus for the life out there. Meaning, if you are under starvation, then it's, from an evolutionary point of view, good to tune down the metabolic system, so to say. And that would increase the possibility for the fetus to survive as a child later on. And then he realized that if these kids then later on were developed during starvation, but later on came into a situation where there were no starvation, then they increased in body weight. And this is a long story, but it's a very fantastic story. So here we have half of the data showing that, okay, we can have low birth weight by chemicals, but can we say something about this shape of the curve, the trajectory curve? And this is a very complicated area, but we have a student, a PhD student, that's been working with this, Catherine Svensson, and I have another friend at Mount Sinai in New York, Chris Jennings, that have been, she's a math person from the beginning. And there is a lot of information out there that growth is a matter of regression model. It's a curve. So if you have these three variables, alpha, beta, and delta, you can really explain how the growth curve looks for nearly all living things on this planet. It doesn't matter if it's a human being, an animal, a flower, or whatever it is. It's follow this pattern. And we are interested in this then for the kids in the SELMA. And this is a matter of, we are interested in mainly two things. We are interested in the birth weight, but that is another story. But then we are interested in this beta, which is the angel, meaning how high is the peak graft velocity. The more shapy this curve is, the stronger increase, the higher increase in weight. And then we also are interested in this inflection point, namely when the kids are increasing in weight and then start to decrease in weight. And this is in speed. So if we can establish these numbers, then we can look on not single BMI or single weights. We can look on the shape of the curve, which is the interesting thing. And I have here two examples, two kids in the SELMA study, A and B. And we have from the registers, of course, in Sweden, all their weights at different time points. You can see for A and B here. Then what Chris did a very hot summer in 2018 in my garden, she applied this function I showed you before. And it is amazing how the same function can fit so very much into both these kids. As you can see, it means that they have completely different shapes of the curve, but both of them follow the function I showed you earlier. And then we get values. So you can see here for kid A and kid B, we have a beta and we have a delta. So then we can, by this data, we can start to look, okay. And the question here is there any association between what these kids are exposed for and how their growth trajectory curves look like? And Catherine published a paper just a month ago. Here we have some question. Okay. So what she did, you can read about the details in the paper, but what she could see was that you can see here to the left, she looked for birth weight. She looked for this infant growth spurt, means where the peak, the velocity, which is the beta here, how shape was the curve here. And then she looked at the age when this peak in growth velocity happened. And you can see here, she did the analysis overall in boys and in girls. And you can see that there were significant associations. We shouldn't go into details here, but what she could see was that the exposure in week 10 of pregnancy of these 26 chemicals, different kind of mixtures of them, was associated to how the shape looked like. And the point here is that then we have, if we go back to David Barker, you can see here that we have biochemicals looked at this birth weight point, but now we also start to have evidence that the exposure during early life, where David Barker showed that this is a matter of starvation. Now we can say that there are chemicals in the environment that can make the same story. And this is based then on real observational data. And we can see that both birth weight, both the shape of the curve in terms of how the infant growth spurt rate, how it looked like in terms of speed, and also when this inflection point was. So what David Barker showed for starvation, we now start to learn could also be a matter of environmental chemicals. And that means that I would interpret this as this is a matter of programming, much more than toxicity, which is the point with these kind of chemicals. So then my last part of this is about SELMA as a platform, as I said. I will show you a slide here, which is a matter. Let's see here, I have two more. Yes, I had I don't know, I think you can see the question here from Maria Noring. From a regulatory perspective, one needs to more or less prove exposure from different chemicals articles. Have you in some way looked at how the mothers were exposed to the different chemicals? Well, we have measured these chemicals in the body of the mother in urine and serum, but you may mean that if we have looked on sources. Is that correct? You can talk if you want.
Hi, yes, that's what I mean.
Yes, and we had a dissertation just 1 month ago, no, in mid-December, Anna Sofia Pries, that have done exactly this. She have followed chemicals from the sources that leak into the environment, and when the chemicals are in the environment, they have been taken up by humans, and then we can relate them to different kind of outcomes. So we call that principle a full chain model, and this is a very good thesis. And Anna Sofia Pries, you can find her thesis on the net. So and if you don't find it, you can send me an email. I will send it to you, because exactly what you say, this is a very important question. So this slide is trying to explain how we now try to use the SELMA study, and I will focus on these 4 cubes in the middle here. 1, 2, 3, 4. I mean, risk factors. What I've been talking to you about now is risk factors, meaning which chemicals or which compounds or which diet or what is it that is related to an outcome that we don't want to have. That is a typical and the old way of looking on environmental epidemiology. 95% of all epidemiology is about that, period. And that is one of the reasons that we don't very often use environmental epidemiology for risk assessment. What we are trying to build up now is to go further. Because one of the important things, if we find an association between, for example, a chemical in early life and an outcome later on, is why is it? What is the biological mechanism behind that? And this is almost always a matter for experimental toxicology in cells and animals. But we have started now to provide data that we can get information on this very important problem also from epidata. The next thing, which is a growing story and very important, is to not only focus on risk factors, but also on resilience factors. And I will show you a little bit on a very typical resilience factor, namely diet and nutrition. Because that is, I mean, many of these chemicals we can't do very much about, the persistent, for example. But is it like that there are resilient factors that, for example, diet and nutrition that can compensate for bad actors? It's a very complicated question, but we are trying to find out ways here. And finally, I will give you a short introduction to a new method called G-computation, which is a way to do intervention in epidemiology data. You may have heard about the Nobel Prize in economy this year or last year, which was attaching this approach, namely to look on unemployment and salaries and how do you examine that in a society, because you never know. You have the data on salaries, you have the data on unemployment, but what is a counterfactual? It means that instead of asking the question what is, you ask the question what if? If we can we reduce the exposure? Do we see any effect in the outcome then? I will explain it a little bit more in a second. But these four are a way to try to get out more information from epidata. And the first one you have already seen. This is a typical story on risk factor. I told you before, prenatal exposure for BPF was related to a lower IQ in children, a typical risk factor. The next one is about a biological mechanism. Green 2B at the top of this figure is a gene, and methylation is a way to measure how active this gene is. If it's totally silent or if it's active from 0 to 100%. So here you can see we have the BPF exposure to the left, we have the full-scale IQ to the right, which is the outcome. I showed you before that there was an association between BPF and IQ. And here we do a so-called mediation analysis. We are investigating, is it like that this association between BPF and full-scale IQ could be explained by DNA methylation in this gene? The reason why we took Green 2B was that Green 2B is a gene that had been related to brain development, to different kind of brain disorders. And as you can see here, there were associations between the exposure, the environmental exposure, and methylation, This is epigenetics, as you understand. And there was an association between the methylation and the outcome. So the result of this was that we could explain of the association we found between BPF and full-scale IQ. Around 8% of that association could be explained by this pathway, namely methylation of one gene. You may say 8% is not much, but I would say the opposite. How can it's almost impossible that one gene can explain so much here. So there are, of course, bias in these models, but we could see associations here, and we could come a step further to what we call causal inference, because we get an indication of an mechanism for this.
Carl-Gustaf, this is extremely interesting, but we are approaching 50 minutes now.
Yes.
If you could kind of try to round off at least in 3 or 4 minutes.
I will try to do that, yes.
Yeah, great.
So then I go into diet. And diet, as I said, is very much called, looked upon as a resilience factor. We have diet data for the mothers during pregnancy, and we have established an index for diet. And we could see that depending how the mother was eating, she could improve the cognition of the kids when they were 7 years old. So better nutrition during pregnancy improved IQ, and chemicals in for example, this BPF decreased the IQ at 7. So what you are doing in this fourth part, the intervention part, is that can we look upon these two potential, one is an beneficial factor, better food, and the other one is adverse factor, namely bad chemicals. And this can be done with this kind of technique called G-computation. So this picture is showing the result. To the left, you have the WQS index I showed you earlier, namely the mixtures of the chemicals. So what we asked here was, if we reduce the chemical mixture, we reduce it by 1 standard deviation below the mean, then we can see here. So the 0 is the current situation, and the -1 if we reduce the mixture in the data we have. Then we could see that we increased the IQ for both boys and girls. And to the right, you can see that if you increase good diet, meaning better nutrition, from 0 there, you increase it instead. And you can see that you get a significant effects for both boys and girls. So by this, we could say we made an intervention in real world and could see an effect of that later on in the children. So this is my final slide. And instead of just looking to the left in this picture and identify risk factors, which is the common way of interpreting epidemiology and using epidemiology, we, by adding on mechanisms, resilience factors, and not at least this new track of interventions, real life interventions, we may produce data that could be of even more importance for better risk assessment at the end. Thank you.